Hello friends, I am here with my channel Solving Questions with Gauri Ma'am. In continuation with my lecture series, today we will see next 4 amino acids. So starting with cysteine. The 3-letter code is C by S and with single letter we denote it as a capital C. This is the structure of cysteine. This structure, we see that it is having SH group in its side chain. This is a sulfur-hydral group which is present in a cysteine. We have seen methionine in which there was a sulfur, and this is the another amino acid cysteine which is having sulfur-hydral group. So in all there are 2 amino acids having sulfur in their side chains. One is methionine, another is cysteine. Cysteine shows absorbance of UV light at wavelength 255 nanometer. Due to the presence of sulfur-hydral group in cysteine, it forms a disulfide bond. Whenever protein is forming disulfide bond, remember that cysteine must be there. The example is a keratin protein which is present in a hair. So there is a ample of cysteine present in keratin. Yes, moving further with 2 more amino acids, asparagine and glutamine. Both are having same functional group in their side chain, CO and H2. If we see the structure of asparagine, the regular structure of amino acid, then CH2 and this functional group is there. And here one more CH2 is there in side chain, and then there is a functional group. Both amino acids are uncharged polar side chain. The 3-letter code for asparagine is ASN and it is also denoted by capital N. The glutamine is denoted by capital Q and GLN. Asparagine is the amino acid which is found at the end of alpha helix and beta sheets. This is also used in the N-linked glycosylation. N-linked glycosylation is the process takes place during protein trafficking, and to this nitrogen of this asparagine the glycosyl chain is attached. So this is used in N-linked glycosylation. This is taking place in the endoplasmic reticulum. The sequence in that protein must be asparagine X, means any amino acid, or other than proline, and serine or threonine. So asparagine X, serine, threonine, this sequence is used, and to the asparagine residue N-linked glycosylation is carried out. Glutamine acts as a nitrogen donor for the synthesis of non-essential amino acid. This is also important in the nitrogen transport to the liver. The next one is a histidine. This is the amino acid which is having imidazolium ring in its side chain. This is a imidazole group which is present in the side chain of the histidine. The HIS is the 3-letter code and capital H is the single letter used for the histidine. This amino acid is present in large amount in hemoglobin, in protamine, and it is also present in histones. This amino acid can act as a buffer at pH 7. That is nothing but a physiological pH, and that's why this is present in a hemoglobin. Remember this. This is the amino acid which can act as a buffer at pH 7. That is physiological pH, and that's why it is present in a hemoglobin. So with this we have completed 4 amino acids: cysteine, asparagine, glutamine and histidine. In my next video we will cover last 4 amino acid. Till then, everyone. Don't forget to like, share and subscribe for my channel and also to hit the bell icon. Thanks for watching.